Afternoon. Yes. Okay. So let's just wait for a minute and let other schools also join and we'll continue the class. Okay. Okay, chalo. We are good to go. Good afternoon, everyone, once again. Today, what we are going to discuss is we are going to continue the previous class chapter, that is a plant growth. Okay, plant growth and development. This is what chapter we were discussing in the previous classes, right? So we have discussed how plant will grow, like how to know the growth rate of a plant and all the other stuffs we have seen in the previous class. And we have also studied in the previous class that the factors which affect the plant growth, right? So out of those, some factors are like growth hormones, water, oxygen, temperature, right? These are all the some few factors which are going to affect the growth rate of a plant, or which affects the growth of a plant. Out of that, one is plant growth regulators. Yes or no? Do you all remember we have studied this in the previous class? Plant growth regulators. Remember again, regulate. What do you mean by regulator? We all know the fan regulator, right? What the fan regulator will do? What is the use of fan regulator? Just for the purpose of show. No. Then to increase the speed of a fan or to reduce the speed of a fan. Right. So regulator, here also the same thing. What the regulator will do is, yes, what the regulator will do here is like, it is going to either increase the growth of a plant or it can decrease the growth of a plant. Right? Which school it is? Just a minute. Okay. So those things about this plant growth regulators, or we can also call the one more name, which is a phytohormones. Phytohormones. Okay. So phytohormones, hormones, we know hormones either they can increase the work or decrease the work, which is about a growth. Phyto here means a plant. Okay. So plant growth regulators can also called as a phytohormones. Now let us see how many phytohormones are there. Actually, the phytohormones or the plant growth regulators have been divided into a natural phytohormones and a synthetic phytohormones. Natural phytohormones means they are inbuilt in the plant already. Synthetic, where we are creating in the laboratory conditions. Okay. So synthetic, some examples here we have a NAA, 2,4-D, 2,4,5-T. Okay, so we'll study about these things. And in the natural growth hormones or natural phytohormones, we have a plant growth hormones and or plant growth promoters and the growth inhibitors. Okay, so this plant growth promoters are Auxin, Gibberellin, Cytokinin. Growth inhibitors, again I'm talking about a regulator, either it can increase or it can decrease. Increasing in the sense, there are three which are going to increase the plant growth. One is Auxin, Gibberellin, Cytokinin. And there are some hormones which can decrease the growth of a plant. Those are ethylene and abscisic acid. Okay, chalo, let us talk about these 5 hormones in today's class. Especially, we should know about the functions. Okay, so why we should know about the functions is in a general life. Let me just give one example. That is mango, tomato, or apple. If there are some fruits, what we'll do? These fruits, if we want to ripe the fruits, we are going to dip the fruits in some chemicals. No, what are those chemicals actually? These are the chemicals called as phytohormones. Okay, so such many examples are there in our daily life of this Auxin, Gibberellin, Cytokinin, ethylene, and abscisic acid. Okay, so that is why we should know about the functions. Okay. And this slide we have already studied in the previous class, right? Plant growth promoters, which are going to promote the activity like a cell division, enlargement of a cell, tropic growth, pattern formation, and flowering, fruiting, seed formation. Many of the growth activities is going to happen. Those are called as a plant growth promoters. Examples are auxins, gibberellins, cytokinins. Same goes for the plant growth inhibitors, which are going to inhibit the activity like dormancy, abscission. Okay, that is by a abscisic acid and the ethylene. This ethylene usually can be active. Sometimes it is acting as a growth inhibitor or the promoter. Okay, we'll see. First one is a auxin. Before studying about the function of auxin, how we got to know about the auxin? What is this auxin and how we got to know that this is a promoter? By the Charles Darwin. Okay. So this Charles Darwin and his son, who's a Francis Darwin, these two started to work on like some grass called as a canary grass. What they understood is the tip was growing. Okay, so then after some time, what they did, okay, whether the tip is growing or the entire plant is growing, we don't know. What they did is they have given the light only from a one side. Okay, usually light will be from all the sides. No? What they did, they kept that in a dark box and given the light from only one side. Now, this tip starts to grow only towards the light. First tip starts to grow and then the entire plant starts to grow. Then they understood there is a something in the tip which is helping the growth of a plant. Okay, there is something in the tip which is helping in the growth of plant, and that is the auxin. Later they understood. Who did this? Charles Darwin and Francis Darwin. Okay, so later comes F.W. Went, who isolated this auxin. Okay, F.W. Went, who isolated this auxin. Is this clear? Now let us see the functions of this auxin. Before the functions, there are two types of auxins. One is a natural auxin and one more is a synthetic auxin. Synthetic, which are man-made auxins. We have studied already two types of plant growth promoters. Same. In the auxin also, from the plant itself, we can extract the auxin in the form of indole 3-acetic acid, that is IAA, indole butyric acid, IBA. Okay, these two can be extracted directly from the plant or isolated directly from the plant. But the same way, auxins can also be prepared in the laboratory conditions. And those auxins are 2,4-D and NAA, naphthalene acetic acid. NAA means naphthalene acetic acid. Is it clear? Is it clear? The historical background of this auxin, how this auxin has been produced, and where we have seen the auxin mainly in the tip of a stem, right? Mainly we have seen this in the tip. Now, let us see the function of the auxins. What I mean, if the auxin is present in the plant, how it will regulate the growth. That is what the function of this auxin. One is, one is that it initiates a rooting in the stem cuttings. Usually, we'll see the stem cuttings, like especially what hibiscus flower. In the hibiscus flower, we can cut the stem and we can keep that in the soil, and later on we can see the rooting in that. So how does this rooting forms? By the presence of auxin. Okay, and especially in a pineapple, the flowering in the pineapple is by the auxin. So all these are helping in the growth or not? Yes, they are helping, right? To prevent a fruit and a leaf drop at the early stage, auxin helps very much. Okay. And parthenocarpic, to form a parthenocarpic fruits. What do you mean by parthenocarpy? Yes, use the chat box and give me the answer. What is a parthenocarpy? Use the chat box quickly. I can see the chat box. You have to answer for this. Parthenocarpic fruits means what? Yes, I'm waiting for the answer. Let's see who will give the answer. How many schools are there? 8. Nobody? Very good. I got an answer from Jainvi Srikakulam, which is absolutely correct. Parthenocarpic fruits, which means the fruits which are formed without a fertilization, right? So to induce that parthenocarpic fruit, especially in a tomato, also we can add a auxins. If we are just adding auxins to the plant, we can see the parthenocarpic fruits over there, and some herbicides, right? So these are all the uses or the functions of the auxin in the plant. If there is an auxin in the plant, we have to know that the tip of the plant will be growing very well only because of the presence of the auxin. Okay. And if we are having a stem cuttings, and from that stem cuttings, we are going to get the roots because of the auxins. And flowering promotion, promoting of flowers and parthenocarpic fruit formation. Okay. So these are all the functions of auxin. So now can we say auxin as the growth promoter or growth inhibitor? What do you say? Auxin is a growth promoter or growth inhibitor, whether it is a promoting the growth or it is reducing the growth. Yes. Answers again in the chat box. No one is answering. Yes, it is promoting the growth. So this is a growth promoter, auxin. Is it clear regarding the auxin and how auxin has been introduced the first time or discovered the first time in the plants? This was discovered by the Charles Darwin and his son Francis Darwin. Okay, in the canary grass. The coleoptile means, you know, no, coleoptile, coleorhiza in the monocot seeds. In the monocot seeds, let's say this is a plumule and this is a radicle. On the plumule, there are many layers. These are called a coleoptile. Okay, this coleoptile will be grown only in the presence of auxin. And this auxin was first time isolated, or this auxin was isolated by a F.W. Wendt. Okay. Yes. In a higher plants, the growing apical bud inhibits the growth of a lateral buds. It is known as a apical dominance. So, but here one thing is there by this auxin. As we all know, auxin which is going to grow apically. Apically means what? In the tip. In the tip, if the plant is growing, that is mainly because of auxin. But sometimes what this auxin will do, let's say in the tip some buds are coming, at the same time in the lateral side also some buds are coming. But these buds which are coming at the tip, these are going to dominate the lateral bud because of the presence of more auxin here. This is called a apical dominance, the dominance which is at the tip, called as a apical dominance because of the presence of the auxin. Okay, so by removing the shoot tips, which results in the growth of lateral buds, right? So if we are removing this tip part or buds which are at the tip, we can see the growth of these lateral buds now, which means, yes, this is the one, the apical bud is the one which is stopping the lateral bud to form. Okay, this is why, this is because of the presence of auxin at the tip. Okay. Yes, this question we'll discuss. Now, the first one is auxins. About auxins we have discussed. And now let us talk about a gibberellins. Okay. Yes. So these gibberellins are, let me write with the other color. Oh. Just give me a second. these gibberellins are acidic plant growth regulators. These gibberellins are what? Acidic plant growth regulators. First time these gibberellins were identified by the E. Kurosawa. What he did is he identified there were some rice plants. They were affected by the fungus which is called as a Gibberella fusicora. They were affected by the fungus called a Gibberella fusicora. Okay, so they have been affected by this plant, by this fungi. This is a fungi. Then later he understood what is exactly is causing disease. Fungi, okay, fine. But inside the fungi, what is the thing which is causing disease to this plants? Then he identified there is an acid, and that acid is called as a gibberellic acid or gibberellic acid. Understand? And what it was doing is this Gibberella fusicora was causing a disease to the rice plant, and that disease is called as a Bacon disease. Bacon disease. So this is how first time the gibberellic acid or the gibberellic acid has been identified by the scientist called as a E. Kurosawa. Okay, is it clear? This is about a historical background, just nothing. There was a scientist called E. Kurosawa. He identified that the rice plant has been got a disease called a Bacon disease. And how this Bacon disease got? That is because of the fungi called a Gibberella fusicora. And this Gibberella fusicora fungi is causing the disease because it was having an acid called a gibberellic acid. So this is how we got to know about a gibberellins or a gibberellic acid. Okay. But these gibberellic acid are of thousands of types. Sorry, 100 gibberellins are present. Out of that, G1, G2, G3 or GA1. Okay, let me erase it. Yeah. GA1, gibberellic acid 1, gibberellic acid 2, gibberellic acid 3, and so on. Total 100 varieties of this gibberellic acid were there. But why this gibberellic acid should be there in the plant? What is the use of this gibberellic acid? Now let us see. The function of this gibberellic acid is that they increase the length of our axis. Okay, the one example is grapes. We all know the grapes. Okay, so if we are spraying this gibberellic acid to these grapes, what happens is the length of the stalk of the grape stalks will be there. No, that length is going to increase. If the length is increasing, obviously it seems like there are many fruits, and which means if the length is only this much, let's say only length is this much of there, and many fruits will be arranged one beside the other, there are chances like fruits cannot grow properly and fruits may damage, right? If we are spraying a gibberellins to the these grapes, then the stalk length will be increasing and fruits can be placed or grow sufficiently. Okay, and the fruits like an apple, usually we all know smaller apple and bigger apple are there, means most probably will go for the bigger apples only. So for the economical reasons, if we are spraying this gibberellins, the fruit shape will also increase, or the improves, elongated and improved shape fruit of the apple. And they delayed senescence. Senescence means what? Aging. Aging in the plant is called as a senescence. Remember this. This word is usually will be using in this chapter the most, especially botany will be using this. Okay, so if any fruit is being if the fruit is being aged or ripened. Okay, so we don't want the fruit to be ripened because the market value is very low now. So we want the fruit to be ripened after some time. What we can do is we can just spray the gibberellins so that the aging will be delayed. It will not stop permanently, but the aging will get delayed. Okay, and there is a one more, GA3, gibberellic acid 3 will be used in the beavering industries like a wine preparation and all these things. Okay, and one more thing, in the sugarcane, as I said already, it increases the length of an axis, right? So sugarcane, which part of sugarcane we will use usually to make a juice? Anything? Anybody? Which part of sugarcane we will use usually to make a juice? Exactly. Oh, Srikakulam students are very active. Only I'm getting answer from them. Okay, that is a stem, right? So if we spray the gibberellin there, obviously the stem length will be increased. If the stem length is being increased, then we are going to get the more and more yield. Yes or no? So for that also it is useful. And most of the plants, this gibberellins can be used. And bolting, for bolting means what? Bolting is an internodal elongation between the nodes. If we want to increase the space, that for that also gibberellin can be used. Okay, that is just before the flowering. In beetroot, cabbage, okay, and in many plants. Is it clear? This is about a gibberellin. Auxin, where the, especially in the auxin, what we can say is in the auxin, the tip of the stem will be increased. So apical dominance is a major in the auxins, by the auxins. Gibberellins, mostly we have to remember is the stem length or the axis length will be increased. If you want to increase the length of anything, okay, Yasmin. Yasmin wants me to explain regarding the grapes. Okay. Yes, as I was saying the same thing now, the length of the stem is going to increase. Now, let's say we have got a bunch of grapes. Okay. This bunch of grapes are having 25 grapes in that, but the length is very less. Only this much of length is there. To adjust all the fruits in this length is very congested. So fruits may get damaged, or because of the lack of space, fruits cannot grow properly, right? So at that time, before the formation of fruit, if we are just spraying the gibberellins there, the axis will be, the length of axis will be increased. If length of axis is being increased, the fruits can occupy very spaciously and they can grow accordingly. Yes or no? Is it clear, Asmi? Ultimately, gibberellins mostly will be using for the elongation of internodal regions. That's it. Especially in sugarcane or bolting in the beetroot and cabbage and grape stalk. Remember all this. Okay. Is it clear? Auxins and gibberellins, two things are done. Now, cytokinins. Okay. So regarding cytokinins, yeah. One interesting factor is there here. That is, this was first identified by the Spoog and the Miller. Who's that? Spoog and Miller. Okay. So what they did is, in the tobacco plant, the internodal regions were growing, or the cells of internodal regions were growing. Then they saw, or they have extracted the area which was growing. They saw that there was a auxin was present, and or they have extracted those cells and they took the auxin from there. Along with the auxin, what they did is they have added a coconut milk. To that, they have added a coconut milk. Okay, to those cells or the area which they have extracted from. To that they have added a coconut milk or extraction of a vascular tissue. They have added in anything, either that or this, anything we can add, or the yeast extract, or the DNA extract. Whatever we add, by this what they are understanding is the stem starts to grow. Okay, the stem starts to grow. Then they got to know, along with the auxin, there is a one more thing or one more phytohormone is there, which is helping for the growth of a plant. Okay, that phytohormone later they named as a cytokinins. Cytokinins means what? Kinins or kinetin. First, they have given the word as a kinetin, which means division. What is, because the cytokinin, again, I'm telling you, the most important function of this cytokinin is a cell division. First, auxin, apical dominance, gibberellin, internodal elongation. Third one is the cytokinin for the cell division. Okay, so kinetin is for the cell division, means then later they have added a word called a cyto to this. Cyto means for the cytoplasm. So it will be helping in the cytoplasmic division especially. That is why they have named it as a cytokinins. Okay, but there is a speciality in this. That speciality, what always I said you, the cytokinins will be acted or they will be acting whenever they mixed with the auxins, but they will act differently. How? If auxins and cytokinins are in an equal number, where the cell divides continuously. Division will be continuous division. There is no differentiation, dedifferentiation, redifferentiation, nothing will be there. If auxin and cytokinin content is equal, or let us say if auxin content is more than the cytokinin content. If we want, let's say I want to grow one plant in the laboratory condition. First, I want to have only the cell division. At that time, what I have to do? I have to add auxin and cytokinin equal content. Then I want roots to be formed. What I will do? I am going to increase the auxin content than the cytokinin content. Now, root division or root formation starts. And after the root formation, I want shoot to be formed. At that time, I should increase the cytokinin than the auxin. Now, the shoot formation is going to happen. Okay. So, in the first, if auxin and cytokinin are in the equal number, there is no differentiation. But if auxin and cytokinin are not equal, there is a chance of differentiation. Is it clear to every one of you? Is it clear? Cytokinins are mostly meant for the cell division or mostly help for the cell division. Okay. I took a... What is the function of the cytokinins? As I said, it will be helpful in the cytokinesis. Kinesis is the division, cyto for cytoplasm. So they will be mostly helping for the cytoplasmic division and helps to produce a new leaves, chloroplast in leaves, lateral shoot growth, and adventitious shoot formation. All these will happen only by the cell division, right? So if new leaves have to be formed or a shoot has to be grown, adventitious root has to be formed, right? If roots has to be formed means auxin should be more than the cytokinin. Okay. And if we are mixing the cytokinin with the auxin, then we can overcome the apical dominance. Because if there is only auxin, this auxin will be presenting at the tip it is going to dominate the lateral buds. But if the plant is having both auxin and cytokinin, we can overcome this apical dominance. Okay. Is that clear? 3, auxin, gibberellin, cytokinin. Is it clear to everybody? Please let me know in the chat box. Yes, thank you. I can get the very active answers from the students. That's great to see. Okay. Next is one more, that is ethylene. How they got to know about ethylene? Just I'm going to tell a few about very few about a historical background. Okay. First, what happened? They have observed there was some gas which was producing from the ripened oranges. Have you ever observed if we keep the ripened fruit and a non-ripened fruit together, then that non-ripened fruit also will get ripe very quickly? Yes or no? But if we are keeping that separately, it will take some more time than that. What do you mean by this here is every ripened fruit is going to produce the ethylene, and this ethylene is going to increase the age of the plant or increase the senescence of the plant. Or this ethylene is mostly in the gaseous form. Okay, this is a gaseous PGR. Gaseous PGR. Okay. Now let us see the functions of this ethylene. One is it influences the horizontal growth of a seedling, horizontal growth on the ground. Okay. And then it promotes the senescence and abscission. Senescence means what? Aging. Abscission is leaf fall or fruit fall, anything. Because it is reducing the growth here. If plant has to grow, the senescence and abscission should not happen. Here, senescence and abscission are happening because of the ethylene. Okay, and as I said, it will be mostly helpful for the fruit ripening. Remember, auxin, gibberellin, and cytokinins are, all three are the growth promoters. And ethylene, which is a growth inhibitor, it is going to increase the age of a plant. Okay, and it breaks the seed and a bud dormancy. Dormancy means what? There is some particular time period where at that time they cannot grow, either seed or buds, they cannot germinate. So that time is called as a dormancy, seed dormancy or bud dormancy. By adding the ethylene, we can break this seed and bud dormancy. You might be knowing about the seed and bud dormancy. Mostly in our homes, as soon as we get a crop, some of the crops we will not sow immediately. We will wait. Okay, let us keep this for 2 months. Let us keep this seeds for 3 months. Then we can sow immediately. If we are sowing also, we will not get the crop. Like parents will say no sometime. That is because the seeds are having a dormancy. But if we want to break that dormancy, we can add this ethylene. Okay. And it also initiates the germination of peanut seeds, sprouting of potato, many. Okay, this is about ethylene, and you can see the some more functions of the ethylene also. Rapid internodal or petiolar elongation. What do you mean by petiole? Petiole is leaf stalk. The stalk of a leaf is called as a petiole. Okay, petiolar elongation, internodal elongation, all these are going to be happen by the ethylene. Okay, root growth and root hair formation, initiate of flowering. Okay, mostly we are going to use this ethylene in agricultural forms. And those who are growing the fruits in your farm, you might be knowing, we will get the ethylene in the market in the form of a ethiophone. Okay, in the form of ethiophone, we are going to get the ethylene in the market. That is usually, this we are going to get in a liquid form. We'll take this liquid, a bucket of water. In that we are going to add 1 or 2 caps of this ethiophone. then we're just going to dip the fruits, especially mangoes. You might be knowing this. Mangoes will be dipping in that bucket and just will keep on the some dry area. You can close the room, and after some days, after 1 to 3 days, you can see the mangoes are being ripened. This ethiophane is a liquid form or liquid source of the ethylene, where we are going to get in the markets. Okay, as I said already, tomatoes, apples, many. Is it clear? So far we have studied how many? 4 growth regulators we have studied, and the last one is the abscisic acid. Okay. Is it clear for everybody? Please answer in the chat box. Yes, very good, very good, very good. Now the last abscisic acid we will study, that is usually in the year of 1960s, we have got to know there are 3 growth inhibitors. Okay, those 3 growth inhibitors are inhibitor B, and then abscin 2, and then a dormin. These were the 3 growth inhibitors we got to know in the year of 1960s. Later, we understood that all 3 belongs to the, yes, all 3 are identical, and they are known as the abscisic acid. Okay, so and the function of this abscisic acid, and I'm telling you again, this is an inhibitor which will be inhibiting the plant growth and the metabolism of the plant growth. Why it should, does it really requires for the plant? Yes, in the unfavorable conditions, if the plant is growing also, plant cannot get any nutrition because of the unfavorable conditions, right? So at that time, if abscisic acid is there, then it is going to inhibit the plant growth and the metabolism. It also inhibit the seed germination just to escape the unfavorable conditions. Okay, and let's say outside there is so much of temperature and the plant is not at all getting a water. At that time, if the transpiration is happening, so much of water loss is going to happen, and moreover plant is not getting a water. So at that time, transpiration is not at all recommendable for this plant. That is why the closed stomata has to be closed, so that the transpiration will not happen. That stomata will be closed by the abscisic acid again. Okay, and this abscisic acid is called as a stress hormone because it increases the tolerance of a plant to various kinds of stress, like any kind of abiotic stress is there, and for that tolerance will be increased only by this stress hormone called abscisic acid. Okay, and seed dormancy, bud dormancy, okay, all these are also going to happen by the abscisic acid. Is it clear? This is about the abscisic acid. So in today's class, totally we have studied about 5 plant growth regulators. Out of that, auxin, which is, can you tell, auxin, which is present at the tip or at the apex. Gibberellin, especially for the internodes, to grow the internodes, and cytokinin for the cell division, or especially cytokinins. Okay, so these 3 are the plant growth promoters. Plant growth inhibitors are the ethylene and the abscisic acid. Ethylene is in a gaseous form, and abscisic acid is an acidic form which is going to inhibit, especially abscisic acid will be inhibiting the growth of a plant, or it is going to increase the dormancy period. Okay, and it will not allow the seed germination in unfavorable condition. So this abscisic acid is called as a stress hormone. Is it clear to you? Yes. So that's it for today, and we'll meet in the next class. Thank you. Yes, this is called a stress hormone. You can see here. Thank you. Bye-bye.